Hello, welcome. I am Onder Bozdogan. I am from Kırıkkale Üniversity Medical School, Pathology Department. In this session, I would like to talk about electrophoresis with a low power look and the practical points. There are some questions after this session, after this presentation. I think you will get the answers of these important questions, but I am not sure the other questions in the science we cannot get, like is there a life on Mars? Electrophoresis is made from two words. One of them is electro and the other is phoresis. Electro means electric and phoresis called migration of carrier across. It's a kind of separation technique based on differential migration features of charged molecules in an electric field. It's an analytical method frequently used in molecular biology, biochemistry, medicine, and plant. History of electrophoresis of very long. However, first studies in electrolysis and other areas of liquids and electric from Faraday studies. However, when we look at the modern electrophoresis, one name is so important. Is Arne Tiselius is the father of the electrophoresis. He made first modern electrophoresis unit called Tiselius apparatus. Electrophoresis is based on important principles. Principles is very simple. In an electrical field, charged molecule and particles migrate to opposite charge. Usually we do it in aqueous solutions. Due to varying changes, masses, different molecules and particles in the mixture migrate at different steps. As a result, separate into single fractions. We see that as bonds. Main components of electrophoresis is very simple. You need buffers, two anodes which supplied by electrical field by power supply, and sample. Migration depends on several factors. Strength of electric field is very important. If you give high voltage, your molecules speed more. Other important features of environment is temperature and also some important features for the material. Net charge of the molecule, size of the molecule, of course the 3D shape of the molecule is also important. Gel type is important because of the pore size different from one gel material to another. Also concentration is important for pore size. Buffer type is important for specific application, of course. Electrophoresis separates everything that can carry a charge: the nucleic acid, protein, peptides, amino acid, organic acid, drugs, and etc. In molecular pathology, although there are some sophisticated and automated techniques, but we use molecular biology, electrophoresis in some areas. Determining quality of DNA and RNA is covered in other presentations. Analysis of PCR products, mutation detection, south and north and blotting, sequencing. Proteins also we use generally Western blotting for detection of the proteins in the gel. Electrophoresis types, there are a lot of different techniques in the electrophoresis area, but there are gel electrophoresis and the others we can say. Gel electrophoresis includes agarose gel electrophoresis and polyacrylamide gel electrophoresis. The other important technique, pulse field gel electrophoresis for big DNA fragments. Capillary electrophoresis is an automated type of gel electrophoresis. Isoelectric focusing for and 3D electrophoresis is important for protein research and protein detection. Gel electrophoresis, there are some main components. You use a gelatinous material and the gel acts as a support medium. We use electrophoresis, generally proteins and nucleic acids. There are three main gel types, but two is important, agarose and polyacrylamide. We use agarose for nucleic acid, but not so small nucleic acid, and large proteins. However, we use polyacrylamide gel for proteins, small nucleic acid fragments. Agarose gel electrophoresis is an easy, fast, well-established method of separating DNA fragments. Agarose is a polysaccharide and derived from a seaweed. Its formulation is a little bit confused, but it's a sugar, galactose. It easily dissolves in boiling water and becomes gel when hardens or when cooling. Bigger pore size than generally polyacrylamide, but there are some micro pore size agarose in the market. Agarose gel concentration and DNA size is very important area. You can use suitable concentration for your size of your fragments and your PCR products. Polyacrylamide gel electrophoresis, it has advantages, some advantages and some pros and cons for agarose gel electrophoresis. It's a synthetic polymer. It is formed from acrylamide with cross-linker methyl bisacrylamide, and this polymerization catalyzed by ammonium persulfate and TML. It has give some high resolution than agarose gel electrophoresis. However, we must say that acrylamide is a dangerous neurotoxin. This is a table for selecting gel concentration and acrylamide bis ratio. You see very small fragments, 40 base fragments can be separated by polyacrylamide gel electrophoresis. Buffer is an important part of the electrophoresis units, electrophoresis tests. It prevents pH charging because of the hydroxy and oxy gases from the electrophoresis. Two common buffer is important. One of them is trisborate etta and other is trisacetate etta. You can buy commercial or you can prepare these buffers by your laboratory. Equipments of the classic electrophoresis is very small. You need a power supply, it's not so expensive, and cooling apparatus, it's a mechanical or other techniques. Electrophoresis gel apparatus, vertical, maybe horizontal, and you need a UV box and the white box. Also, you can use digital camera gel documentation systems, which is very suitable for electrophoresis studies. Reagent is, of course, there is a gel staining chemicals, e.g. ethidium bromide, prepared gel or gel chemicals for preparing gel, buffers, and loading dyes. Other laboratory equipment, also you need like pH meters, pipettors, lab scales, and stir plates. This is an horizontal gel apparatus. It is very easy to use. You can easily cast your gel in this unit and easily adapts different techniques and technicians like this kind of electrophoresis so much because so easy. Generally used for this kind of electrophoresis apparatus for agarose gel electrophoresis. However, vertical gel is used for polyacrylamide gel electrophoresis. However, it has some leakage problem when you cast gel, but new units and new complicated some instruments and apparatus solve this problem. This is a gel documentation system in our laboratory. It's a computerized system, and you can analyze the bars by computer-based. This is inside of the documentation system. This is UV lamp and the white lamp by seeing the bars easily. This is our supply. Techniques is not so complicated. However, it has some tricks. Please do not forget before casting a gel, DNA is an organic acid, negatively charged and run to anode, positive electrode. If you remember this, you can, or remember this, your DNA will be in the buffer. Small fragments go further than the large fragments of the DNA. Do not forget running off the gel. Do not drink a coffee because you lost gel. You lost your DNA in the buffer. Cut off electricity before taking gel from apparatus. And please do not forget ethidium bromide is a highly mutagenic. On the other hand, polyacrylamide is a highly toxic material. Markers is very important for your electrophoresis test. Treatable marker is needed. If you do not select treatable marker, you cannot easily detect the length of the fragments. Ethylene bromide is an important dye in molecular biology and molecular pathology. It works well. Because of it works well, we know it's a powerful mutation, but we use it's a cheap, sensitive, easy to use, and fast. Binds to DNA. Fluorescence under UV lamp can be directly added to gel when you're preparing, or the buffer, or gel can be stained after run. I prefer add directly to the gel because I think it's stuck on the gel and easily dissolved. Concentration is changed from your applications. There are some ethylene bromide alternatives, cyber grain, silver stain, methylene blue, and commercial stains also found in the market, but they are generally more safe but less sensitive. Cyber stain also has some mutagenic effects on human. Voltage is another important parameter of electrophoresis, classical electrophoresis. But if you give high voltage, low resolution, and increased temperature, as a result, low quality separation. Generally, less than 5 or 8 volts per centimeter length is enough for classical electrophoresis. But empirical approach is more important. Technique. When we look at the technique, here we focus on the agarose gel electrophoresis because it's simple and you can use it in several areas. Technique, first you need a sample. Sample may differ from one test to another. You may get PCR products, DNA cut with restriction enzymes, and the others, proteins, and etc. This is our laboratories. This is the equipment and the reagents for use simple agarose gel casting. We have got a microwave oven, a power supply, and gel apparatus, TBA buffer, tank concentrate, agarose, and distillate water. You first weight the agarose desired concentration. Its concentration change. I use 2 concentration because it easily handled. Dissolve in buffer, swirl the solution periodically, and melt in microwave. Clear solution you can get when melt, and there is no particles in the solution. Be careful, the boiling agarose solution may be so hot. After waiting a few minutes, nearly 60 Celsius temperature, you need ethylene bromide, and you need also insert the comp in the apparatus unit. And pour the gel. Wait until the gel hardens. 20 minutes is generally required, but it is related to the temperature of your room. Pull out the comb carefully. Please, this is an important area because you can tear the gel easily. Be careful. And do not forget, ethylene bromide is a very mutagenic, and wear gloves. And after this, first, you need load markers. Generally, markers came by bromophenol blue. However, it changed from another. If it has not loading dye, you must mix with loading dye. Be careful not to contaminate the other wells when loading the samples, and do not perforate the wells. Loading dye includes bromophenol blue generally and glycerol. Glycerol used for the sinking of the material, and bromophenol blue for the tracking of your electrophoresis. Running of the gel, control the anode and cathode. This is an important point because before you change the red and black cable from to another, then power source is turned on. Look at the air bubbles. Air bubbles shows you your gel is running. The time depends on current and the concentration of the gel. Please track the bromophenol blue. Generally migrates 1.5 concentration. Agarose is near 300 paspa. Then take off the gel from the unit. Stain if you do not add lithium bromide before the gel casting. The gel is then visualized by UV light. You can analyze the gel. You can also cut the bands, the gel for further techniques. For example, plasmid technique. You can get the DNA from the gel and remove the gel for southern blotting and the protein for western blotting. This is one of area I use electrophoresis so much, primer selection. If you say, if you began to a new study, you need a new primers. This is 5 primers here. Excuse me, 1, 2, 3, and 4 primers. This is primers, dimers. This is not so good, of course. This is unexpected bond, maybe related to DNA contamination of the primers detect another area in the DNA or messenger RNA sequence. I select this because there is no primer dimer. Only there are a few very weak bonds here. However, we can solve this problem changing the annealing temperature. Generally told, real-time PCR, quantitative real-time PCR, do not use electrophoresis, but generally I use after real-time PCR. I make electrophoresis because, like here, this low degree contamination is easily seen. Protein electrophoresis is a simple to use and a highly electroprotective technique. Provide information of molecular weight, charge, subunits, and purity of the protein mixture. STS page is commonly used technique here. There are mainly 2 main type of technique for protein electrophoresis, also maybe DNA. Native charge is the molecules is a natural position. However, you can denature gel electrophoresis by this separate folded proteins by their size. STS polyatomic gel electrophoresis is a kind of denaturing electrophoresis. Give information of the size of the polypeptide change. Separated length of the polypeptide, not it is a charge. This is a classical STS page gel. 2 gels, stacking gel and separation gel. Stacking gels important is for concentrating proteins and different formulation, and the separating gel for resolve the bands. Polyacrylic gel electrophoresis is the first step of the STS page is the first step of the Western blotting. Acrylic concentration and molecular weight scale. Proteins also stained by some stains, chromasil blue dye, silver staining, and the other fluorescence is important area for this. There are other electrophoresis techniques, however, we only look at it. Isoelectric focusing is a technique generally detect the isoelectric points of the proteins, and 3D page is 2-dimensional electrophoresis. First, you separate by PL, isoelectric points, and then by size in a mixture. It's 2 is important area of proteomics, and Western blotting is Gaia will present the importance of the Western blotting in molecular biology next session. And the first step of Western blotting is, of course, STS polyacrylamide gels. Full-speed electrophoresis is used for very large DNA molecules. The anode and cathode change periodically, and the large molecules can run. It's a gold standard for epidemiologic studies, for example, E. coli subtypes. Capillary electrophoresis, which pointed before, is an important area of gel electrophoresis. It has several advantages, but it's an expensive technique. The instrument is very expensive. Capillary electrophoresis is based on classical gel. You have buffer, sample, and the capillary tube. Capillary tube has gel-like material, and detector collect the data, and you can get an electropherogram and easily analyze. But we said that before, it is expensive. Capillary electrophoresis, you can use several materials: urine, serum, DNA, drug screening, pesticide, food contents, etc. In molecular biology and in classical routine, you can use capillary electrophoresis in neoplastic disorder, tumor-related mutations, mucosal instability, analysis of molecularity, and the other genetics areas you can use. At the end of my presentation, I would like to say electrophoresis also is an old-fashioned technique, but it is useful now. It is easy, it is very cheap, and you can shoot at least a horizontal electrophoresis in your molecular laboratory. This is my home aquarium, reef aquarium. Thank you for patience.